Welcome, everyone. I'm just going to wait a minute for everybody to join. Okay, so welcome everybody to our HEALEY platform weekly webinar. We have a special webinar this week. We've invited one of our industry partners, Denali Therapeutics, to come and talk to you about the science behind their drug, which is Regimen G, and to be available to answer your questions. So I want to thank people who come weekly, who are here again, and then welcome the new people. I'm Cudkowicz. I'm an ALS clinician and lead of the HEALEY ALS Platform Trial, and I'm also here with Dr. Suma Babu, who's a neurologist and an ALS scientist at Mass General Hospital, and is one of the leaders of Regimen G, or the Denali arm of the HEALEY ALS Platform Trial. And with that, I know you all came to hear about the science of Regimen G, and I want to introduce Dr. Dana Jennings from Denali Therapeutics, who we've gotten to partner with and really enjoy working well with, who's going to tell you more about their drug and why they're excited to bring this forward to people with ALS. Dana.
Thanks so much, Merritt, for the kind introduction, and thank you all for joining us today. I want to just take a couple minutes and introduce my other team members that are joining me today. So I have Deepika Tamali, who is leading the clinical operations for this program. I also am joined by Fen Huang, who is the translational scientist and works on our biomarkers. And Leslie Kane, who is a biologist and works in discovery, and Kim Ramsey, who is an advocacy specialist and works with our advocacy group. So without further ado, I am going to get started. So again, I'm Dana Jennings from Denali, and it is clearly my pleasure and to provide an introduction to DNL343, which is a new investigational drug being developed for the potential treatment of ALS. And we're very excited that DNL343 has been selected as Regimen G for the HEALEY platform trial. There we go. And by way of disclosures, I just want to point out that I am an employee of Denali Therapeutics, and DNL343 is a drug that is under investigation and has not been approved by any health authority, and such as the FDA or the European Medicines Agency. So I'd like to start by introducing Denali Therapeutics for those of you who may not have had the opportunity to work with us. Denali is a small biotechnology company with headquarters in South San Francisco. And our somewhat ambitious mission is to defeat neurodegenerative diseases through rigorous therapeutic discovery and development. Now, some of the diseases that we're focused on are shown here and include lysosomal storage disorders, which are rare genetic conditions and that have multiple neurological manifestations, Parkinson's disease, Alzheimer's disease, frontotemporal dementia, and then of course the condition that we're here to discuss today, which is ALS. So at Denali, we're committed to some key scientific principles as part of our development programs, and we believe that this will really increase our likelihood of success in developing drugs to treat these devastating neurological conditions. So just starting on the left, now, there have been major advances in our understanding of genetics, which has provided some key insights into abnormal biochemical pathways that contribute to the onset and progression of many or multiple neurodegenerative conditions. And each of the programs that we lead at Denali is really aimed at addressing a genetically defined pathway. So secondly, in the middle, our approach includes ensuring that we deliver our therapeutics to the brain. Now there's a specialized system that protects against the passage of toxic substances to the brain, and this is called the blood-brain barrier. And this actually can present some substantial challenges in effectively getting drugs to the brain. So Denali scientists have developed state-of-the-art technologies to ensure that our drugs, both large and small molecules, can effectively pass through the blood-brain barrier and reach the targeted abnormalities in the central nervous system. And finally, our drug development programs are driven by data obtained through biomarkers. So biomarkers are molecules that are found in the blood. They can be found in other body fluids or tissues as well. And biomarkers are particularly helpful in determining how well the body responds to a treatment and for selecting individuals that are most likely to even respond to a treatment. So very valuable in drug development. And so our overarching goal of this scientific approach is really to bring more effective therapies to individuals living with neurodegenerative diseases, including ALS. And we find it incredibly valuable and helpful to learn directly from individuals and families living with ALS, and as we strive to develop these more effective treatments. So through consultations and ad boards and so forth, we've gained some valuable insights from individuals with ALS that have really helped inform our clinical trials, ensuring that our trials are actually feasible for participants. So we're highly committed to making a meaningful difference to those living with ALS, and we currently have 2 active ALS development programs in clinical studies. So DNL343 is an eIF2B activator with the potential for reducing TDP-43 pathology, and I'll be reviewing the mechanism of action for this compound in the coming slides. And DNL343 is actively being evaluated in our ongoing phase 1b study and also in the HEALEY Platform Trial as Regimen G. And we'll be discussing again this in more detail here today. So SAR 443820, also known as DNL788, is a RIPK1 inhibitor that is targeting an inflammatory pathway. And for this compound, Denali is partnered with Sanofi, and Sanofi is leading the phase 2 Himalaya study in participants with ALS. So what is the mechanism of action for DNL343? Now, the remainder of this presentation will be focused on DNL343, and I'd like to begin by describing the pathology or changes in the nerve cells that occur in ALS and our therapeutic hypothesis regarding how DNL343 may address these pathological changes. So the discovery of many ALS-associated genes over the past decade have provided evidence that there is a link between stress granule biology and changes that occur in the nerve cells in ALS, including abnormal clusters of a protein which is called TDP-43. Now, around 95% of people with ALS have been shown to have accumulation of abnormal clusters of TDP-43, and this interferes with normal cell function. And TDP-43 is widely recognized as a key pathological feature of ALS, and clusters of TDP-43 accumulate in the cells during conditions of stress. So interestingly, TDP-43 can be found in structures called stress granules. And many genes that are linked to ALS are part of stress granule-related pathways. So individuals with ALS, stress granule pathways may play an important role in driving the accumulation of harmful TDP-43 and ultimately the degeneration of the nerve cells. So now that I've introduced sort of the importance of stress granules and TDP-43 pathology in ALS, I'll now describe how this ties into the therapeutic hypothesis for DNL343. So the overarching therapeutic hypothesis for DNL343 is that it protects against an abnormal stress response. And there's now evidence that when the cells of the body are stressed by certain conditions, such as infection or trauma or environmental factors, or certain ALS-associated genes, this can initiate an integrated stress response, or the ISR for short. Now, the ISR is a process that triggers inactivation of EIF2B, or eukaryotic initiation factor. As part of the ISR, normal protein synthesis is interrupted and stress granules are formed, which may facilitate the formation of abnormal TDP-43 clusters that are seen in nearly all forms of ALS. And ultimately, this leads to neurodegeneration. Now, there's also an increase or activation in stress response biomarkers, including ATF4 protein and CHAC1 gene expression, which can serve as biomarkers to demonstrate whether or not we are actually impacting the ISR pathway with our therapeutic. So how does DNL343 impact the ISR? So DNL343 is a small molecule that inhibits the ISR pathway by activating the EIF2B. Now, activation of EIF2B results in the cells returning to make normal amounts of protein. The stress granules ultimately dissolve, and there's a reduction in TDP-43 clusters. This has the potential for restoring normal nerve function. And in addition, the stress response biomarkers are also reduced. So what evidence do we have that DNL343 inhibits the stress response? So this slide briefly reviews an experiment conducted using cells in a petri dish and evaluating the effects of DNL343 on stress granules and TDP-43 clusters. So this figure really nicely shows that when cells are exposed to a chemical stress, stress granules form, concentrating a key stress granule protein, which is shown here in red, and TDP-43 shown here in green in the cytoplasm. And the overlap of the stress granules and the TDP-43 is also shown here in yellow. So when DNL343, with the addition of DNL343, these stress granules dissolve over time, and the TDP-43 clusters also reduce. So the takeaway from this experiment then is that the EIF2B activator DNL343 reverses both preformed stress granules and TDP-43 clusters. So we also tested the effects of DNL343 after an acute or short-term injury in healthy mice. Now, a common experiment for testing whether a therapeutic can protect against an acute injury is through pinching or crushing the optic nerve cells in the retina. So crushing the optic nerve cells activates the ISR, resulting in retinal cell death. So this graph shows that DNL343 decreases the integrated stress response in the retina. Now, on the y-axis, we show the level of CHAC1 gene expression, and on the x-axis, the level of treatment with DNL343. The gray bar is the control, and the white bar represents the effects of the crush injury with an increase in CHAC1 gene expression. Now, in mice that were pre-treated with DNL343, there's a more limited stress response following the crush injury, and nearly a normal level of ISR biomarker was observed. And the graph on the right here shows data regarding the survival of the optic nerve cells 14 days after the injury, and compares those that were treated with DNL343 prior to the injury with, again, the control population. And so the two highest doses of DNL343 had nerve cell survival that was very similar to that of the control. So DNL343 decreases the integrated stress response in the retina and reduces cell death in mice. So we've also completed some highly informative experiments using an EIF2B loss of function mouse model. Now, these mice have low EIF2B function and are therefore highly relevant for evaluating DNL343 since it activates EIF2B, and therefore one can test whether it actually improves function in these mice. So EIF2B mice have a hyperactive ISR, or integrated stress response, in the brain that causes nerve cell death and ultimately motor impairment. So in addition, they also demonstrate an increase in the stress response genes, including CHAC1 gene expression. Now, when these mice were treated with DNL343, there was a clear reduction in CHAC1 gene expression and showing a reduction in the integrated stress response with increasing doses. And this approached basically the normal levels at the highest dose. And in terms of motor function, we also measured the time it took the mice to cross a balance beam to evaluate for the improvement in motor function with DNL343 treatment. Now, the EIF2B mice typically take twice as long to cross the balance beam due to their motor dysfunction, which is shown here. However, with DNL343 treatment, there's an improvement in function that basically results in normal function across the balance beam, again, at that highest dose level. So DNL343 decreases the integrated stress response in the brain and protects motor function in the mice. So what is our clinical experience with DNL343? So shown here are the study designs from our phase 1 study in healthy participants and the phase 1B study in ALS participants. And there were 95 healthy participants evaluated in the phase 1 study and 27 ALS participants in the phase 1B. Now, the healthy participants completed once daily oral dosing with DNL343 for up to a 14-day treatment period, while the ALS participants completed oral daily dosing for a 28-day period. Now, it should be noted that the ALS participants were also offered an 18-month open label extension that remains ongoing. Now, the main goals for these early phase studies were to evaluate the safety and tolerability of DNL343, evaluate the levels of DNL343 in the blood and in the spinal fluid, and to measure the effect of DNL343 on the ISR biomarkers to determine if we're actually inhibiting the stress response. So what were the characteristics of the ALS participants in our phase 1B study? In terms of demographics, we had 83% that were male, 17% were female, and the median age at enrollment was 57 years. It should be noted that the sex and the age of the participants was comparable across all treatment groups. And in terms of the Ellis-Gorriel diagnostic criteria, 24% were classified as definite, 59% probable, and 17% as probable with lab-supported data. And there was a higher percentage of participants with definite ALS in the low-dose group in this study. Now, the median duration from symptom onset was 22 months, and there was also a shorter duration from onset in that low-dose group. So we measured the levels of DNL343 in the plasma of healthy participants and ALS participants, which is shown here. So across both populations, there's an increase in DNL343 concentration levels with increasing dose levels. And on the x-axis is the time from dosing. And you can see that after that initial increase in exposure, the level remains pretty steady over the 24-hour period. And this demonstrates a relatively long half-life supporting once daily oral dosing for DNL343. And I also want to mention, although the data is not shown here, that the levels of DNL343 were also measured in the cerebral spinal fluid, and they very closely mirror the unbound levels measured in the plasma. So this means that the drug DNL343 is able to pass through that blood-brain barrier and reach the nervous system where it is most likely to have an impact on the nerve cells affected in ALS. So we also evaluated the impact of treatment with DNL343 on two ISR biomarkers that I've mentioned previously, which are ATF4 protein and CHAC1 gene expression. These biomarkers were measured in a component of blood, which is called PBMCs, or peripheral mononuclear cells. And in healthy participants, there was a substantial reduction in ATF4 protein of greater than 60% with DNL343 treatment, and a similar level of inhibition in the ALS participants in both the low dose and the high dose. Likewise, DNL343 treatment resulted in a robust reduction or inhibition in CHAC1 gene expression, which you can see here with a reduction of greater than 60% with a similar level of reduction in the ALS participants. So these studies, in these studies, we observed robust reduction of two ISR biomarkers, providing evidence that treatment with DNL343 is actually inhibiting the integrated stress response. In terms of tolerability in the healthy participants, DNL343 was generally well tolerated. There were no serious adverse events, and the majority of adverse events were mild in severity. The most common adverse events listed here: headache, fatigue, and dizziness, which were more frequent in the DNL343 treated participants compared to those with placebo. And myalgia, muscle spasms, nausea, and the increased CPK were less frequently reported. So there were two discontinuations. Both were considered by the investigators to be unrelated to DNL343 treatment. One was a personal circumstance, and the second event of anxiety. And for the ALS participants, during the 28-day double-blind portion of the study, DNL343 was also generally well tolerated in this population. There were no serious adverse events, and the adverse events were of grade 1 or grade 2, so mild to moderate in severity. And the most common adverse events were headache and fatigue, which occurred at a higher frequency in the DNL343 treated participants compared to the placebo, but were all in the mild category. And less commonly, some of the participants also experienced hypogeusia, which is a reduction in the sense of taste, and pre-syncope, or near fainting, and nausea. And there was one participant in this study that discontinued due to a rash. So the key takeaways from these studies are as follows. So DNL343 is administered through once daily oral dosing, which is supported by the long half-life based on the pharmacokinetic profile. There's extensive distribution of DNL343 to the cerebral spinal fluid, and the biomarker data from the healthy and the ALS participant studies show that DNL343 inhibits the integrated stress response. And thus far, DNL343 has been generally well tolerated in both healthy and ALS participants. And data from these phase 1 studies continue to support further development of DNL343 for the potential treatment of ALS. And we're really delighted to have the opportunity to evaluate DNL343 as a Regimen G in the HEALEY Platform Trial. And enrollment for Regimen G began in May of this year, and it remains ongoing. So this graphic provides a very high-level summary of the study design for Regimen G. The screening period is 42 days, and approximately 240 participants that are eligible will be randomized to either DNL343 or placebo in a 3-to-1 ratio. So essentially, for every 4 participants that are enrolled, only 1 will be randomized to placebo. And the double-blind placebo-controlled treatment period is going to be 24 weeks. There's also an active treatment extension, which is a 52-week minimum. So the goals for Regimen G are as follows. First is to evaluate the effect of DNL343 compared to placebo on ALS progression. So we really want to address this question: can DNL343 reduce the progression of ALS? And our second goal is to evaluate the safety and tolerability of DNL343 in a larger number of participants with ALS with chronic daily dosing. And then finally, to evaluate the effect of DNL343 on select biomarkers, including biomarkers in the cerebral spinal fluid. So I want to say just a few words about cerebral spinal fluid collection, or CSF collection in general, and then provide some details regarding CSF collection specific to Regimen G. So in adults, there's approximately 120 milliliters of CSF that surrounds the brain and the spinal cord, and it's constantly being replenished by producing about 20 milliliters per hour. And CSF collection is actually completed by inserting a small needle into the lower back below the level of the spinal cord and withdrawing the spinal fluid. So for Regimen G, we'll be obtaining approximately 20 milliliters of CSF at baseline and in addition at week 24, and this will be used for biomarker testing. So CSF is one of the most valuable fluids to evaluate biomarkers for ALS, since it has a direct contact with the nerve cells and in the brain and the spinal cord that most closely reflects the changes that occur in the central nervous system. So what biomarkers will we be measuring in the CSF? First, the levels of biomarkers impacted by the integrated stress response will be measured to determine how DNL343 is impacting this stress response. And we'll also evaluate biomarkers assessing the impact on the nerve injury or neurodegeneration, which will include, but not limited to, NFL, the neurofilament light. And while CSF collection is optional for Regimen G, we really hope that all participants will consider providing CSF given the valuable information that will be gained from analyzing these biomarkers. So on behalf of Denali and the DNL343 team, I want to thank the individuals and families for generously contributing to our clinical studies. And I would also like to express our gratitude to the clinical sites that have helped us provide the valuable data in our early phase studies. And we are very much looking forward to continuing to work with the ALS community toward the development of DNL343.
Thank you, Dana. We have a lot of questions already, which is fantastic. I wanted to just remind everyone that we are recording this and we will be posting it, the recording on our website like we do for all the regimens. So if you want to go back and hear it again or hear certain parts of it, it will be available. So Dana, I might ask you the questions, but obviously if you want to direct them to anyone on your team, please do. So there's a question about one of the exclusion criteria in the HEALEY ALS Platform Trial for Regimen G, which is a mention of a diagnosis of epilepsy or seizures. And they just wanted to understand that a little better, if you could explain that.
Yeah, sure. So this is a great question and certainly one of interest to us. So in our preclinical studies and animal studies, of course, we test much higher doses of the drug, and in those studies, we did see in the mouse model that there was some tremulousness and potential, movements or the convulsion-like activity that actually led us to be very conservative, because now, of course, we're testing much lower doses than what we did in the animal models. Certainly, in our early phase studies, in the phase 1 and the phase 1b studies, in the human studies, we did not see any of this activity so far. So, I think, the better part of caution is to make sure that we don't have people that are at risk for seizures, but we don't anticipate that there would be any issues going forward at the doses that are used in the clinic.
Thank you. There's actually been some epidemiology studies suggesting that epilepsy is less common in people with ALS. And so I haven't seen very many, if anyone, with that. So I don't think this is really going to exclude many people. So a related question is whether there are any drug-drug interactions with anything, any exclusionary type meds.
Yeah, so there are a number of medications that are exclusionary, mainly because we're still testing the drug-drug interaction with DNL343. So those, I believe those are probably listed in clinicaltrials.gov, but you can certainly go to clinicaltrials.gov and see those. But certainly, as we do additional clinical pharmacology studies, we anticipate that list will get shorter, and we don't, again, trying to be cautious in these early stages of development in terms of the potential, I would say, drug-drug interactions.
Thank you. There's a question about whether Denali is testing DNL343 for vanishing white matter disease. There's someone who notes that some of those preclinical results were shared at the NEALS meeting in 2021.
I don't know if Leslie or somebody else on the team wants to take that question. Of course, we're interested in vanishing white matter disease. I mean, I think right now our focus is pretty much entirely on evaluating DNL343 in ALS, but we do have an interest certainly in understanding vanishing white matter disease and how our drug might work there.
Okay. There's a question about whether the neurons that are being targeted by DNL343, are they the upper motor neurons and the lower motor neurons, or just one or the other, or do we know?
I mean, I think it's a really interesting question. So, we've focused, a lot of our work on looking at, the central nervous system and the upper motor neurons, but certainly we would anticipate that this would have an impact on both the upper and the lower motor neurons. It wouldn't differentiate between the upper and the lower motor neurons.
Yeah, it's a good question. A lot of our models, tend to look more at the lower motor neuron, though I know Hyundai and others are developing upper motor neurons. But we do know that the TDP-43 biology is in both upper and lower motor neurons, so it would make sense that this drug would target both. There's a question in your phase 1b. Did you determine DNL343 target engagement also in the CSF or just in the blood?
Yeah, so for both the phase 1 and the phase 1b, we measured DNL343 in the CSF, and it again, the level in the CSF pretty closely mirrors the unbound plasma level in the blood. So we are getting really good exposure in the CSF with DNL343 exposure.
Thank you. Suma, this question might be for you. It's more about the platform trial in general, but the question is, do the 24 weeks from the double blind begin from the day I start the trial, or do I have to wait until the 240 spots fill?
Thank you for that question. So the 24-week really starts for each participant from the time that they enroll in the study. You don't have to wait for all 240 spots to fill. So once you enroll and you meet all the eligibility criteria and you get to the baseline visit, and that's the starting of the 24-week period.
Thank you. There's a question about combining your two ALS drugs. So since chronic type 2 inflammation can cause stress, did you try in animal models the combination of DNL343 with DNL788?
Yeah, we have, I don't know if Leslie or somebody else wants to take this. I'm not working on the 788 program, so, uh-oh.
Yeah, so far as I know, I don't think there's, we have tested them in combination. I think there's sort of different models that we test the two in because, as Dana showed earlier, the primary model where we've done a lot of mouse work in this context is that EIF2B loss of function model. And I don't believe that we've tested the RIP kinase inhibitors sort of in combination in that model.
Yeah.
It's a really interesting idea though.
Mm.
Suma, maybe you could answer this question also. It's about when do we expect results from this trial for the platform trial of Regimen G?
Yeah, so Merit, maybe I'm going to need a little bit of guidance from you here. As the regimen has just started, so people are starting to enroll. The sooner all 240 participants are able to enroll, and then we wait until the last participant completes the last visit, and then there'll be a database lock, and we look at the results and present the results. So if I were to project the timeline, Merit, would you say maybe next year, sometime in 2024?
Yeah, I hope so. I think it's all going to depend on enrollment rate, and we have started many, but not all of the sites are activated for G. We're working very hard to get all of them activated, and there's a lot of interest. And again, we thank you guys for coming on this call and spreading the word and talking to your neurologist about whether this is a good trial for you. But we're hopeful that it'll enroll fast, high quality, and we'll get the results as soon as possible. There's a question about neurofilament, and that since it's getting more attention now in the field from the FDA and others, did you look at this in your phase 1B? And if you did, can you talk about it yet? And then we can also add about what we're doing in the platform trial with neurofilament.
Yeah, Fen, do you want to take this? I can start by saying, the 28 days, we don't think is long enough to actually see any effect on a neurofilament. So, I mean, and it's also a relatively small sample size, but Fen, maybe you want to expand on that.
Yeah, I think, Dana, you basically nailed it. Neurofilament certainly is in the hot seat right now, and everybody wants to see if the drug impacts neurofilaments. And we are excited, but again, as Dana just said, the phase 1B trial is a very small sample size trial, and also with a short duration, and we don't think that the study is powered to detect a neurofilament change right now. But we'll certainly explore that, and we haven't really done the work yet.
I'll add that in the platform trial, we're asking people to donate blood and spinal fluid so we can measure neurofilament and other biomarkers. So there's a question about whether there's potential for DNL343 for people with PLS or primary lateral sclerosis or just pure upper motor neuron disorder.
Yeah, I think that's a fantastic question, actually. I mean, I think, the mechanism of action is really based on stress granule biology and, reducing that integrated stress response. May not be, necessarily only effective against TDP-43, but, could be expanded to include other neurodegenerative conditions, and PLS would certainly fall within that category, I would think. So, stay tuned. I mean, there may be some additional we may expand, the indications that we're interested in evaluating.
Thank you. There's a question about dose, whether you think a higher dose, could be even more effective, or if you're at that sweet spot, the highest possible dose.
Yeah, I don't know that we're at the highest possible dose. I mean, we tend not to try to go into the clinic with our highest possible dose, but I think that we're at a dose that, definitely reduces the integrated stress response biomarkers, or inhibits the integrated stress biomarkers to a sufficient degree, greater than 60%, and in some cases, greater than 80%. So, I mean, I think that that's, we think that that's, a reasonable level to, to expect a clinical benefit.
Thank you. There's just a comment from someone who said that their family member started yesterday at Vanderbilt, and just thanking you for allowing them to participate. And thank you for you and your family member being part of this study. There's two questions that are kind of related, so I'm going to combine them, which is, do you expect to see fast changes in people, or do you think this is a drug or a mechanism that might take some time to show efficacy?
Yeah, this is always a really great question. And I mean, of course, pathology doesn't start overnight, right? So, I mean, I think, just as we don't expect any changes in NFL in 28 days, I mean, I think it's also true that it may take some time. It may take months to actually see a benefit from this drug, and that may be true. I don't know if that's true across all mechanisms, but across many of the mechanisms, I have a feeling that would be true in ALS. I mean, I remember Tim Miller at one point making a comment, after we were looking at the NFL data for tofersen, and he said, it just takes time to heal. It takes a long time for people to ultimately, develop these changes in the neurons, and it also, to reverse it it's just not overnight. So I think we have to set our expectations and be a little bit patient and understand whether we can make a difference with these medications.
No, I agree. I think especially neuroprotective drugs like this just might take some time, and we saw that with the every drug that's on the market took several months to start to see the benefit. So this is a really good question, and glad that it's a hard one. So it's going to Dana, which is, what are the weaknesses in the hypothesis? It's a good question for everything. Are there, any?
So what are the weaknesses of the hypothesis? So, I mean, I guess, we don't know for certain, the impact that the stress response has on TDP-43 pathology and how that is, totally linked together. So that's, I would say, potentially one of the limitations. But I'd also love to hear from Leslie on this question. I think she may have some additional thoughts.
Yeah, I agree, Dana. I think, unfortunately for ALS and for frankly most of the neurodegenerative diseases, we just don't have great animal models that we feel really recapitulate all the aspects of disease. And so as much as we can test our drugs in cell lines and in vitro and in human neurons in a dish, in the absence of a good model of disease, there's always going to be things that we can't really answer until we can actually test it in the clinic. And I think that, to Dana's point, we have great hypothesis and great connections between the integrated stress response and the genetics of ALS and the integrated stress response and stress granules and TDP-43 biology, which we know is involved in ALS. But until we can actually, test it in these trials to really understand, that is going to be the mechanistic hypothesis. And I think it's a good one. And I think it's going to be very exciting to test and see where it goes. But I think that I agree, Dana, that's the primary point.
Thank you. The next kind of related is, like, do you know whether the dose that you've chosen sufficiently inhibits the ISR in the brain? I think you addressed this a little bit, but maybe you could go over that again.
Yeah, I don't know, Fen, if you want to touch on the biomarkers in the brain.
Yeah, the clinical dose was selected based on both our preclinical and clinical biomarker data. So we do have a very extensive data package in our preclinical models to demonstrate that the peripheral inhibition of ISR directly predict the CNS inhibition of ISR just by PK/PD modeling. And also we leveraged our phase 1 PBMC ISR inhibition data and also the CSF PK. So we do predict that the dose we selected will sufficiently inhibit ISR in the CNS in our patient trials.
Thank you. I am at the moment not seeing other questions, but we'll give another minute or two if people had other questions they wanted to ask our colleagues at Denali.
Well, I have to say, these are really fantastic questions. People are really, paying attention to the data. I'm really impressed with the level of questions and appreciate it because, again, it helps us understand and think more broadly about the program. So this is really terrific.
I would just add to that, this is such an engaged audience and great questions. And I must say that Regimen G, the DNL343 regimen has been really exciting. And there's a lot of interest among patients already. We just started enrolling, a month ago, and we have already enrolled close to 30 people as of today, which is very, very exciting. So it's moving very fast in terms of the enrollment.
Okay, let me just check if we've gotten everything. Yeah, there's just a nice comment about thanking us and that their family member is considering enrolling. So again, thank you for that. Well, with that, I'll just remind that we will be putting this up on our website. It usually takes us a day or two to get that posted. We will be inviting Denali back in future ones to talk to you again and answer any other questions. But I want to thank everybody for joining today and thank Dana and the entire team at Denali for coming today as well. Thank you.
Thanks,
Have
everyone.
a good rest of your evening. Okay. Bye-bye.